In our last chapter, we talked about genetics. We talked about Mendel and how big B and little b coded for brown eyes or blue eyes or being tall or short or having a widow's peak. And so in this chapter, we're going to take that information and we're going to put it together to talk about, okay, so how does having big B, this mysterious big B, actually code for you having brown eyes? Where's the connection there? And that link is going to be this, the substance of DNA. So the first thing we're going to do today, we're basically going to talk a little bit about history. So we're going to get into, in our next couple of lectures, we're going to talk about the structure of DNA, how it actually makes proteins, which is what DNA's job is. We're going to talk about how DNA copies itself and how it gets passed on from parents to offspring. But first, today, we're going to talk a little bit about the history of DNA. So the history is actually fairly recent compared to a lot of other discoveries. You know, we knew about cells back in the 1600s, but it wasn't until the 20th century, 1928, when our first inkling about DNA came about. And so the first experiment we're going to talk about was done by somebody named Frederick Griffith, and he studied bacteria that cause pneumonia. So pneumonia, if you don't know what that is, it's a disease where it can be caused by bacteria and viruses. In this case, it was a bacterium he was studying that caused pneumonia. But it causes lungs to fill up with fluid. One of the symptoms of COVID in some people can be that they end up with pneumonia. So it can be deadly in some cases. And what he found was that these bacteria that cause pneumonia, he was studying them causing pneumonia in mice, and he found that pathogenic ones, which are the ones that actually cause the pneumonia, when those pathogenic ones were killed and they were mixed with live bacteria that were harmless, the live bacteria turned into pathogenic ones. In other words, bacteria that were perfectly harmless suddenly could cause pneumonia. So let's talk a little bit about how he did this. So the bacteria had two different coatings. The smooth coat bacteria, these were the ones that actually caused pneumonia. They were what's called pathogenic or disease-causing. So if you look at the picture here, these smooth bacteria, they had this extra smooth coating here around them. That's where sometimes they were called S-strain bacteria. You might see it written as that, S-strain. But they had this smooth outer coating. And for whatever reason, that coating helped them to become pathogenic. When you inject them into mice, sadly, the mouse dies of pneumonia. Well, there were these other bacteria, non-pathogenic, harmless, and they were sometimes called R-strain for rough. And they actually had a rough-looking outer coat. So that's where this rough and smooth is coming from. But the bottom line is the rough-coated ones, if you injected them into mice, or even if you had injected them into a person, nothing happens because they're harmless. So then what he did, he took it a step further and he took these smooth-coated ones and he boiled them to kill them and injected them after they were dead into the mice, and the mice lived. And that proved, because that's kind of an important step, that proved that dead bacteria don't cause disease. That the bacteria, when they're dead, are not capable of making the mouse sick. But the weird part was he took some of these smooth ones that were dead and mixed them with the rough ones, the ones that don't cause disease, that were still alive, mixed those together and injected it into the mouse, and the mouse died. And so what basically had happened, what he had discovered, is something called transformation. And transformation is where a bacteria picks up something from its environment, which he did not know what it was they were picking up. Now, you know, spoiler alert, what they were picking up was the DNA from the dead ones. So in essence, what was happening was you had these bacteria, and the dead bacteria, their DNA was floating around, and these live bacteria absorbed it, and since the dead bacteria's DNA coded for this smooth coat that allowed them to cause pneumonia, the rough bacteria actually turned into smooth bacteria, which would be really cool. Like if you could just drink somebody's DNA and then turn your eyes blue because you wanted blue eyes or whatever. It doesn't work that way in people, but in bacteria it does. Now, he didn't know about DNA. He just made a hypothesis that there was a factor that must be able to turn the harmless bacteria into harmful bacteria, but he didn't know what that factor was. Now, there was another experiment that repeated his. It was done by Avery. They did what he did, but they were able to take the bacteria and sort of isolate the different components, like the carbohydrates, the phospholipids, the DNA, all the different bacterial parts, and figure out that it was actually the DNA. That was the part of the bacteria that was inside the rough ones that turned them smooth. So that's another experiment. We are not going to talk about that particular one, but you can read about it. So the next experiment we're going to talk about is Hershey and Chase. So Alfred Hershey and Martha Chase, they actually studied viruses. Not just viruses, they specifically studied what are called bacteriophages or phages, which are viruses that actually attack bacteria. A lot of people think of viruses as things that infect us only, but there's actually viruses that infect plants. There's viruses that infect bacteria. Now, the ones that infect bacteria, phages, they usually look something like this. There's going to be a drawing on the next page as well, but they're kind of interesting looking. So they only have two parts, and this is what made them a great choice. They talk about by choosing their model carefully. Let me explain what they mean by that. Viruses only have two parts. They have a protein coating called a capsule or a capsid, and then inside they have their nucleic acid. In this case, it was DNA. So they knew when these viruses infected a bacteria and the bacteria died, what they knew was one of these two things has to be responsible for that. And they were able to design an experiment where they could figure out which of these two things it was. See, the thing is with the bacteria in the first experiment making the mice sick, bacteria have all kinds of parts, right? They have DNA, they have phospholipids, they have carbohydrates, they have proteins. So it would be very difficult, especially back in the early 20s when that first experiment was done, to figure out which one of the parts of the bacteria was actually responsible for making the mouse sick and changing the other bacteria. So they just knew it was a factor. This experiment, by using a bacteriophage, we don't have all those different things. There's no lipids, there's no carbohydrates. It's just protein, DNA. Now, how did they figure out which part was getting injected? Well, that's actually a very interesting experiment. So they used radioactive isotopes. Radioactive isotopes, they actually glow under X-rays, and these two radioactive isotopes will glow different colors. So they used phosphorus 32. It's written like this, P32. And it turns out that DNA contains phosphorus, but protein doesn't. So this radioactive isotope would only stain or incorporate into the DNA. And then they used another radioactive isotope, sulfur 35. This also glows a different color. Well, I believe this one glows yellow. And the sulfur 35, turns out protein has sulfur in it, but DNA doesn't. So by using these two isotopes, all they had to do was expose the virus to the isotopes, make sure that the isotopes got incorporated so that there was phosphorus in the DNA and sulfur, radioactive sulfur in the protein, let the virus infect the bacterium, and then look inside and see which radioactive isotope ended up inside. And then they would know which one of these two, protein or DNA, was responsible for causing infection. And so this is a very simplified version of their experiment. So when they used sulfur 35 in the protein coat, you see that they did not find any radioactivity. The radioactivity stayed outside, showing that the protein was not the part of the virus ending up inside the bacteria causing illness. They repeat the experiment with phosphorus 32, staining the DNA, and guess what? They find the radioactivity inside. So they were able to prove that DNA was the substance that caused the virus to be able to infect the bacteria. That was what was causing the infection. And like I said, another scientist did something similar with, he didn't use radioactive isotopes, but he was able to do an experiment using the work from that mouse experiment to show that DNA was also the substance in the bacteria that made the mouse get pneumonia. So they were later able to also show that. And this is just a more in-depth look at the experiment that they did, but you're not going to be tested on the very specific details. All right, and here's a real quick video of it if you'd like to see sort of an animation of that experiment.
In the Hershey-Chase experiment, bacterial viruses, called phage, were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did not interfere with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing P32 labeled deoxyribonucleotides. This resulted in a phage population with P32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the P32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.
Alright, and so that's actually where we're going to stop today. You guys in class, you know that you had an activity where you were going to look at 6 different scientists and their experiments and learn a little more about the history of DNA. And then tomorrow, next time, we're going to go into the actual structure of DNA on a molecular level, and we'll talk about how it's a double helix and how it's made out of nitrogen bases, and we'll get into all the details.